Welcome to the deep dive. Today, we're diving into the world of structural variants in the human genome.
Ooh, structural
Yeah,
variant.
so we're talking about like big chunks of DNA that can change who we are and how our bodies work.
So not just like tiny typos.
Right, more like rearranging chapters in a book instead of just changing a comma.
I like that analogy. And you know what's crazy? Every single human genome has tons of these structural variants.
Really?
Yeah, they're part of what makes us unique. They can affect our health and even make us more or less susceptible to certain diseases.
Wow, that's a lot to unpack.
It is.
So for this deep dive, what's on the agenda?
So first we got to figure out how scientists even classify these things.
Okay,
Then we'll look at the crazy ways these variants can happen, like from simple copying errors to, really complex DNA stuff.
okay.
And then finally, we'll touch on how often these changes pop up and how technology is helping us to find even more of them.
Sounds good. Let's start with the basics. Like, how do scientists even classify these structural variants?
Well, it's kind of like sorting Legos. You might start by separating them into bricks and plates and wheels.
Right.
It's similar with structural variants. Scientists have come up with categories to organize this huge amount of genomic data.
Okay, so what are some of these basic categories?
One big one is called canonical structural variants.
Hmm?
These are your everyday deletions and duplications.
Deletions and
Yeah.
duplications.
Think of them like missing or extra Lego bricks in your DNA.
Gotcha.
They're the most common types, and their size can vary a lot from a few base pairs to millions.
Okay, so deletions and duplications pretty straightforward.
Yeah.
But I'm guessing it's going to get more complicated.
Oh, yeah, you bet. We also have complex structural variants, and these are where things get really wild. Imagine taking those Legos and not just adding or removing bricks, but like totally rearranging them, flipping them around, swapping them between sets.
Whoa, like DNA gymnastics.
Yeah. Exactly. It's not just simple cut and paste errors with these complex ones. They can involve multiple breakpoints in the DNA and things get shuffled and flipped around. It's no wonder they've been hard to find and study, especially in those parts of the genome that are full of repetitive sequences.
Okay, so it's not just like one simple mistake. It's a whole mess of changes.
Right.
Are all of these complex structural variants equally common?
Well, the good news is that the most extreme examples, like something called chromoanagenesis, are super rare.
Chromoanagenesis?
Yeah, it's like imagine a huge earthquake that shatters and rearranges entire continents. That's
Wow.
basically what happens in chromoanagenesis. Dozens of breakpoints and multiple chromosomes all jumbled up in one big event.
So like chromosome rebirth.
Exactly.
Yeah.
But thankfully, it's super rare. We're talking like less than 1 in 10,000 people.
Okay, good to know.
Yeah, it's been seen in some kids with severe developmental issues, but thankfully it's super uncommon in the general population.
All right, so you mentioned that these structural variants can happen in different ways. What causes these DNA puzzles to get messed up in the first place?
Think about copying a really complicated Lego structure.
Hmm.
Sometimes you might accidentally skip a step, add an extra brick, or even swap pieces around.
Right.
Similar things can happen when DNA is being replicated.
So it's like human error, but at the DNA level.
Yeah, basically. DNA replication is super complicated, and even with all the checks and balances, errors can still slip through. Sometimes a polymerase, the enzyme that copies DNA, might mess up and make an extra copy of a repetitive sequence.
So that would be a duplication.
Exactly. Or sometimes breaks in DNA strands might get repaired the wrong way, which can lead to deletions, inversions, or even more complex rearrangements.
Sounds like our cells have a lot to keep track of.
Oh, they do. And all the different ways these structural variants can form are incredibly diverse. But the good news is that scientists are getting better at understanding these mechanisms. They're like DNA detective, looking at the sequence and the context of the breakpoints to figure out what happened.
So like forensic science, but for DNA.
Exactly. For example, if they see blunt ends at the breakpoints with no matching sequences, it suggests a process called non-homologous end joining, where the broken ends are just kind of glued back together.
Makes sense.
But if they see short stretches of matching sequences called microhomology, it points to a different repair mechanism where things get a bit more complicated.
So by looking at the clues, they can figure out how it happened.
Right.
So we're starting to understand how these variants form.
Yeah.
But what about how often they happen?
Ah, that's where things get tricky. Figuring out the exact rate of these de novo structural variants, the ones that appear for the first time in a person, has been a challenge.
I can imagine.
Yeah, early studies could only detect large changes in copy number, and they estimated that these variants showed up in maybe 1–3% of people.
Okay.
But then whole genome sequencing came along and completely changed our understanding.
Whole genome sequencing where they read the entire DNA sequence.
Yep. And with this technology, we can see a much wider range of structural variants and in much greater detail.
Cool.
Yeah, studies using whole genome sequencing suggest a rate closer to 0.11 to 0.29 de novo structural variants per generation.
Wow, so that's like a tenfold increase.
It is. Technology is playing a huge role in revealing the true extent of structural variation in our genomes.
So it's like we're just starting to scratch the surface.
Exactly. And even those estimates might be low because the most common type of whole genome sequencing still has trouble with highly repetitive regions.
Repetitive regions.
Yeah, and that's where a lot of structural variants, especially tandem repeats, to hide.
Tandem repeats are those repeating patterns like in wallpaper,
You
right?
got it. Imagine trying to put together a jigsaw puzzle where a bunch of the pieces look almost the same. It's tough.
Yeah, I can see that being a problem.
But luckily, there's a newer technology called long-read sequencing that can read much longer stretches of DNA. And that's helping us tackle those tricky, repetitive regions.
So it's like upgrading from a magnifying glass to a high-powered microscope.
Perfect analogy. As long-read sequencing becomes more common, we can expect those estimates of structural variant mutation rates to keep changing and becoming more accurate.
Sounds like an exciting time to be studying this stuff.
Yeah.
So we've covered how structural variants are classified and how they form. But let's zoom out a bit. How are these variants distributed around the world? Are they random, or do they follow certain patterns?
That's a great question. And the answer is, as you might expect, complicated.
Figures.
While structural variants follow a lot of the rules of population genetics, they also have their own little quirks.
Quirks.
Yeah, for instance, the number of structural variants you have can actually depend on the technology used to detect them.
Wait, so the same person could have totally different structural variant counts depending on how their genome was analyzed?
Yep, that's a real challenge.
Hmm.
And it's one reason why comparing data from different studies can be tricky.
I can see how that would be a problem.
Generally, studies using long-read whole genome sequencing find more structural variants, sometimes more than double, compared to studies using the more common short-read technology.
Wow, that's a big difference.
Yeah, and that's mostly because long-read sequencing is better at handling those repetitive regions.
So it's not really a fair comparison if the technology is different.
Right. But even with those differences, some patterns are starting to emerge. We know that most structural variants are rare, meaning they're found in less than 1% of the population.
Okay.
And since we've only sequenced a tiny fraction of people globally, we're probably missing millions of rare structural variants that are out there.
Millions.
Yeah, especially from groups that haven't been well represented in research.
So we're really just seeing the tip of the iceberg when it comes to the diversity of structural variants across humanity.
Pretty much. But even with what we know, we can see that most of the structural variants in any one person are actually common variants.
Common variants?
Yeah, the ones found in at least 1% of the population. It seems kind of counterintuitive, but it shows that both rare and common structural variants are important in shaping our genomes.
So we have a mix of rare, unique variants that make us individuals and common ones that we share with others, like a genetic mosaic.
Exactly. And this mosaic is influenced by things like population history and natural selection. Just like with other genetic variations, we see differences in structural variant frequencies across different ancestral groups. It reflects our shared history and migrations.
So deep. So structural variants aren't just telling us about our individual uniqueness, but also about the history of human populations.
Absolutely. And when we look at how natural selection has shaped these variants, things get even more interesting.
Okay, you've got me hooked. How does natural selection come into play?
Well, structural variants can have a big impact on our genes and how they're regulated. Some can disrupt genes directly leading to loss of function or changes in gene expression.
Okay.
Others can affect non-coding regions that control when and where genes are turned on or off.
So these structural variants can be like genetic switches, flipping genes on or off.
Exactly, and depending on the effect, natural selection can either favor or weed out these variants.
So it's like survival of the fittest, but for structural variants.
Right. For example, there are some cool cases of structural variants that have gone through positive selection, meaning they provided some advantage and became more common in certain populations.
Give me an example. I love these evolutionary stories.
A classic one is the amylase gene AMY1, which helps us break down starch. Populations that eat a lot of starch, like those who have relied on agriculture for centuries, tend to have more copies of this gene because of duplications.
More copies.
Yeah, their ancestors who could digest starch better had a survival advantage and passed those extra copies down through generations.
So you're saying my love for bread and pasta could be thanks to my ancestors.
It's possible. We see other examples too, like deletions in the haptoglobin gene that are associated with lower cholesterol or complex inversions linked to more babies in some populations.
So these structural variants are driving evolution.
They are. But it's important to remember that most are actually subject to negative selection, meaning they're bad, and are gradually removed from the population over time. It's a constant balancing act between the variants that are helpful and the ones that are harmful.
A genetic tug of war.
Exactly.
So with all this talk about structural variants being linked to diseases, does this mean that anyone with one is going to get sick?
No, not at all. It's super important to remember that not all structural variants are pathogenic,
Hmm.
meaning they cause disease. Many are benign and just contribute to the normal variation we see in people.
So it's not like a guaranteed bad thing.
Right. The effect of a structural variant depends on a lot of things, like the specific genes or regulatory elements involved, a person's genetic background, and even their environment.
So context is key.
Exactly. The same structural variant might be harmless in one person but cause problems in another.
Okay, so how do doctors figure out which structural variants are benign and which ones are pathogenic?
Well, that's where things get tricky. It's not always easy to figure out, especially when we're dealing with variants in non-coding regions or in those repetitive parts of DNA that are hard to study.
Like solving a puzzle with missing pieces.
Yeah. That's why clinical interpretation of structural variants takes a lot of careful work. Doctors look at the patient's symptoms, their family history, the exact location and type of the variant, and whether it's been seen before in people with similar conditions.
So they're playing detective again.
Right. They also use databases of known pathogenic variants and algorithms to help them figure things out.
So it's like a combination of science and clinical experience.
I got it.
This is all fascinating. We've learned so much about structural variants already. What else is there to explore?
Well, even with all the progress we've made, there's still a lot we don't know. One of the biggest challenges is fully understanding the structural variants hiding in the most complex and repetitive parts of the genome.
Those trouble spots.
Yeah, they're like the final frontier of genomics.
The final frontier of genomics. Sounds exciting and kind of scary at the same time. What's stopping us from figuring out these complex regions?
Well, the main problem has been the technology. Our good old short-read sequencing, although great for lots of things, isn't so good at accurately mapping and assembling those super repetitive parts of DNA. It's like trying to put together a puzzle where all the pieces look basically the same.
So we need a new tool for the job.
Exactly. And that's where long-read sequencing is coming in. These technologies can read much longer stretches of DNA, so we can finally figure out those complex and repetitive areas.
So long-read sequencing is like opening a new door to understanding structural variants and how they affect our health.
Absolutely. We're finding new structural variants, uncovering hidden variation in tandem repeats, and getting a more complete picture of how much structural diversity there really is in the human genome.
That's amazing. And it's not just about finding them right. Long-read sequencing is also helping us to build these things called pangenome graphs.
Pangenome graphs, right. Imagine a reference genome that isn't just a single line of sequence, but like a whole web of interconnected paths. Each path represents a different possible arrangement of DNA segments.
So it's like a choose-your-own-adventure book for genomes.
Yeah, that's a great way to think about it. You can follow different paths depending on the specific combination of structural variants in a person.
Wow, that's so cool.
Yeah, and these pangenome graphs are changing how we analyze and interpret genomic data. They let us take into account all the complexity of structural variation and give us a more accurate picture of human genetic diversity.
It's like a huge step forward in personalized genomics.
It is. And as these pangenome graphs become more complete and widely used, they'll have a big impact on things like medicine, evolutionary biology, and population genetics. But let's get back to the basics for a sec. We've talked about how structural variants arise and how they're spread out in different populations. But now I want to talk about something really important. How these variants actually affect our genes and how they're regulated.
Okay, let's get into the details. How do these big pieces of DNA mess with our genes?
One of the main ideas here is dosage sensitivity. It's all about how a gene's function changes when its copy number changes. We've known for a long time that some genes are haploinsufficient, meaning that having only one copy instead of two causes problems.
So it's like a recipe where you absolutely need 2 cups of flour and using only one just won't work.
Perfect analogy. For years, the focus has been on finding these haploinsufficient genes, especially when looking at developmental disorders. But there's more to the story.
Oh.
Yeah, recent research has shown that some genes are also triplosensitive, meaning that having an extra copy 3 instead of 2 can also screw things up.
Triplet sensitivity. So too much of a good thing can be bad when it comes to genes.
Exactly. It makes sense, though. Genes work together in complex networks, and changing the amount of one gene can throw everything off balance.
Like a delicate ecosystem where every gene has its role and messing with the numbers can have a ripple effect.
Right.
So how do scientists figure out which genes are dosage sensitive?
It's not easy, and it involves a lot of computational work. Scientists look at huge data sets of structural variants and look for patterns of whether certain genes are more or less likely to have deletions or duplications.
Okay.
If a gene is rarely seen with a deletion or a duplication in the general population, it suggests that those variations are probably bad and are being selected against, which indicates dosage sensitivity.
So they're looking for clues to find these sensitive genes.
Yep. And all this work has led to the development of fancy metrics and models that can predict how likely a gene is to be haploinsufficient or triplosensitive.
So we're starting to understand which genes are most vulnerable to these changes in copy number.
Yes, and these maps are really useful not only for basic biology, but also for doctors.
Speaking of doctors,
Yeah.
how does all this knowledge about structural variants and dosage sensitivity actually help in diagnosing and treating diseases?
The impact of structural variants on human health has been known for a long time, ever since they discovered that trisomy 21 causes Down syndrome. And since then, tons of studies have linked structural variants to all sorts of diseases, from common ones like schizophrenia and autism to rare genetic diseases.
So structural variants are involved in both diseases that run in families and those that just seem to pop up.
Yeah. And they play a particularly big role in developmental disorders. In fact, chromosomal microarrays, which are specifically designed to detect large copy number variants, are now routinely used in genetic testing for kids with unexplained developmental delays or birth defects.
So if a child is showing signs of a developmental disorder, doctors can use these microarrays to look for big pieces of DNA that might be missing or duplicated.
Exactly. And a lot of times these copy number variants are de novo, meaning they weren't inherited from the parents, but happened for the first time in the child.
Oh, wow.
Yeah, these de novo copy number variants have been linked to autism spectrum disorder and other developmental conditions.
It's incredible how technology can help us find these genetic changes and maybe give some answers to families who are looking for explanations.
It really is. And as we learn more about structural variants, genetic testing to find them is only going to become more common. But it's important to remember that not all structural variants are the same. Some are pretty simple, like deletions or duplications, but others, as we've talked about, can be super complex with multiple breakpoints and rearrangements.
Right, those complex structural variants, do they also cause diseases?
They do. While they're much rarer than simple deletions or duplications, they can have huge effects, especially when it comes to developmental disorders and rare diseases. They can lead to gene fusions, mess up regulatory elements, or even create entirely new genes with unpredictable results.
Sounds like they could really shake things up in the genome.
They can. And one of the most promising ways to study them is, you guessed it, long-read sequencing. It's letting us explore those hidden parts of the genome where many complex structural variants and repeat expansions are found. We're already seeing some amazing discoveries, and I'm sure long-read sequencing is going to keep changing our understanding of the human genome.
This is all super interesting, but let's think about the individual. What if someone gets their genome sequenced and finds out they have a structural variant? Should they be worried?
That's a good question, and it's something that comes up a lot in genetic counseling. It's important to remember that not all structural variants are bad. Many are harmless and just part of the normal variation we see in people. Figuring out if a structural variant is causing problems is complicated and involves looking at many factors like the person's symptoms, family history, the exact location and type of the variant, and whether it's been seen in other people with similar conditions.
So it's not a simple answer. It's about looking at everything and carefully weighing the evidence. This has been a really insightful deep dive into the world of structural variants. What are some of the key things you want our listeners to remember?
The most important thing to remember is that structural variants aren't just minor players in our genomes. They're a powerful force shaping who we are affecting our health and driving our evolution as a species. And as we continue to learn more about them, they will undoubtedly change how we approach healthcare, understand our past, and shape our future.
It's amazing to think about how far we've come in understanding these hidden variations in our DNA.
It is, and this is just the beginning. As we keep exploring the world of structural variants, I'm sure we'll uncover even more fascinating things that will change how we understand ourselves and how we approach healthcare.
It really is amazing to think about how these changes in our DNA can have such a big impact on our lives. But with all this talk about the future of medicine and personalized genomics, I'm curious about what hurdles we still need to overcome before structural variants are fully integrated into healthcare.
Hmm, that's a good question. I think one of the biggest challenges right now is figuring out what a structural variant actually means clinically, especially the ones that don't directly mess with genes. We talked about dosage sensitivity, but there are lots of other ways structural variants can affect how genes are regulated and how they work.
So it's not just about a gene being missing or duplicated, but how those changes might affect all the complex interactions between genes.
Right. A structural variant could change the location or accessibility of regulatory elements, or disrupt the 3D structure of DNA, or even create these weird new fusion genes with unpredictable effects. Interpreting these things requires a deep understanding of both genomics and molecular biology.
So we need more research and more teamwork between scientists and doctors to really understand how to interpret structural variants.
Absolutely. And we also need to figure out how to standardize the way we detect and analyze structural variants. We talked about how different technologies and algorithms can give different results, which makes it hard to compare findings between studies and clinical settings.
So it's like trying to talk in different languages.
Yeah.
We need a common language for structural variants to make sure things are accurate and consistent in clinical practice.
Great analogy. We need to develop standardized protocols and reference databases. So structural variant testing is reliable and understandable no matter what technology or lab is used. It's like setting up a common set of rules so everyone's playing the same game.
That makes sense. So what's next for structural variants? Where do we go from here?
Well, on top of those challenges with interpretation and standardization, I think one of the most exciting things is to develop better models of structural variant mutation rates. We've touched on this a bit, but it's about going beyond just figuring out overall mutation rates. We need to develop models that can predict exactly where in the genome these structural variants are most likely to pop up.
So it's like having a map that not only tells you the general area, but also gives you the exact address.
Exactly. That level of detail is super important. It will help us understand how structural variants form, predict what they'll do, and develop better statistical methods to analyze them in disease studies. Ultimately, this kind of detailed understanding is going to be key for using structural variants in personalized medicine.
It's amazing to think about how much we've learned about these variations in our DNA. What a journey it's been.
It really has. And we're just getting started. As we delve deeper into the world of structural variants, we're bound to find even more amazing things that will change how we see ourselves and how we approach healthcare.
It's an exciting time to be alive and see how the story of structural variants in the human genome unfolds. Thanks for being our guide on this incredible journey.
It's been my pleasure. And to our listeners, keep exploring, keep asking questions, and keep diving deep into the fascinating world of genomics.
That's a wrap on this episode of The Deep Dive. Until next time, stay curious.